Welcome back. You guys just keep coming back for more. So hopefully you had candy break like I just did. So right now it's before Halloween. And now I have a ton of these little guys that I'm totally addicted to, stuck in my teeth. Yeah, there's a ton right there. So I'm going to be sucking on it while chatting you guys up. So by the way, I am addicted to these things. They don't like me so much, and they give me wicked indigestion. So now it's a race against time between, can I finish this lecture before I have a gastric attack? We'll find out. I'll keep you posted. You'll know because I'll be like, so I'm in the same spot I was for lecture 1. This is chapter 12, lecture 2. I'm kind of looking around because you guys are listening to me, but you're probably like, oh, what else is there? And I see my little dog back there watching me. I see my little Planet Mobile, my TV. So you can see some Halloween decorations. But, I mean, there's actually a real lot of Halloween decorations all over the place as I decorate for myself. Where is it? Oh, I can't get you guys up to see them all. Yeah, there's like wacky stuff, scary spiders and whatever. Nope, none. Oh. I have this one decoration. Look at you can close up. So this is on the kitchen table, and it's like a Halloween-y snow globe. You can tell I want to do this lecture, right? And it plays creepy music, and there's little bats. So the game when the kids were little would be to see who could get the most bats landing on the roof of the mansion. So you would flip it upside down and then set it down. And I have like 4, but now I'm moving it. 1, 2. Ooh, I got like 6 or 7 over there. It's not be our little game. High tech. That was before they found Nintendo and everything. Oh. Yes, I am cleaning Twizzler out of my teeth. Sorry. And I'm going to do it again when I'm done with this lecture, but in the next lecture. All right, so what I want to say about this lecture is this lecture is going to hurt. This lecture is technical. We're going to go into some cellular mechanisms. So if you can't handle that, if you're mentally not with me, then don't listen to this right now because you won't like it. This is going to be very cellular in nature. As we're going to dive into a neuron and how does it work. So we had in our last lecture, lecture 1, the layout of the nervous system, the anatomy of a neuron, what are the different parts, what do they do? Cleaning out Twizzler. Sorry guys. I can't even laugh because you'll see all the Twizzlers stuck in my teeth. And then the neuroglia. So let's continue on and make them tiny. If they just take me off. And we're going to talk about the electrical signals. And remember, electrical signals is all about, ions. No, I didn't really. All right, so this is going to be a lot of ions. And they're doors, they're gateways. They're gateways into and out of the cell. And how do they work and how do they flow? And why do I need a constant supply of oxygen and glucose to constantly be making ATP for neurons? It's all because of one little guy. All right. Oops. Electrical signals. So what does this say? Electrical signals in neurons are 2 ways, are 2 different types, not 2 ways. There are 2 different types of electrical signals in a neuron. There's the action potential, which is the impulse, which will go the distance, or there's kind of local little changes of ion concentrations inside and outside of a cell, and that's called a graded potential. And that's just a short little distance, really just within a neuron. Not even enough to do anything, but graded potentials can be additive and then they can be mighty, right? Get their mightiness in numbers, as opposed to an action potential, which is the real deal message shooting along either to the brain, in the brain, or away from the brain, right? Sensory neuron, interneuron, motor neuron. And it all depends on what's going on with ions, right? And so ions are important. And what is this little guy? Oh. Yuck. I don't know that I even really want to talk about this. What's with all those weird little numbers? It must be part of this little thing. Well, here's the deal. So here's a guy. Where are you? Where's my pen? Okay. So here's this guy, and he wants to write. He wants to hold a pen like I'm holding a pen. Hopefully he does a better job writing than I do. So in order to hold the pen and do what he needs to do, the first thing that's happening is I have the sensory receptor, this number 1, on this sensory neuron. So it's sending information toward the central nervous system. And is it showing me? Oh, here's his cell body. Remember how I said the cell bodies of sensory neurons are outside of it in those little clusters, those little blobs? That's a ganglion. All right. So, and that's the dorsal root ganglion. Okay. So what this thing is saying is there is a graded potential happening kind of in the finger by the sensory receptor, but when it's strong enough, then it becomes an action potential, a message. That's this black arrow shooting toward the central nervous system. Then it's going to zip. What is that? Oh, just an interneuron in the central nervous system. And now I'm on the purple guy. It's going to hit one part of the brain called the thalamus. That's the grand central station. Routing, go up to my sensory cortex in the parietal lobe. And then, hey, I actually want to be writing, so I then have to zip along, which is not showing me. I don't know why it's zipping from graded potentials, but get a strong enough signal having the response of a motor neuron coming out of the central nervous system, and motor neuron will ultimately talk to muscles, effectors. These are my skeletal muscles, and I'll hold the pen and write. So it is crazy. When you want to be writing, you have to have all that input, and it's much more complicated than what it's showing in here, because there's all this evaluation. Hey, is he holding the pen too tight? Is he not holding it? Is his writing clear? How is it? It's zipping in a whole lot of different parts of your brain to be assessing the continuous process of writing and constantly adjusting and moving around that pen. It's pretty cool. I do miss meeting you guys in person to just really emphasize how amazing everything we do is and complicated, and we just take it for granted. All right, so before a neuron does anything, and he's not doing anything, he's kind of resting. So I will have a resting membrane potential. And the resting membrane potential will have a difference of ion concentration of different ions across the plasma membrane. And this difference will also lead to a voltage difference. And that means I'll have more positives on one side and therefore more negatives on another side. So I have that in existence, and we're going to be a lot more specific about this in a minute. And then I also have these ion channels. And ion channels, remember, are those integral proteins that span the plasma membrane and make these ion channels. But ion channels are there's a bunch of different types. And when ion channels are open, ions can move. And when ion channels are closed, they cannot. So we have 4 different types of ion channels that exist. And they all exist in neurons in one place or another. So I can have leakage channels. Leakage channels are usually open a lot. Where's my pen? Okay. All right, let's see. Randomly open and closed, but they're open a lot. And the potassium channels I have a lot of leakage potassium channels, which means potassium can pretty much move. You know what? Let's take a break for a second. I'm not going to pause the recording, but let's just say that my major players of ion players, are sodium, Na, and potassium, K. And these guys are frenemies. And really, sodium's kind of stupid. He doesn't know that potassium's his frenemy, but potassium is like, oh yeah, you're here, I'm leaving, right? Potassium's kind of snotty and never wants to really be around sodium, all right? So we want to keep that in mind, that they're frenemies. You think they get along because they both have pluses, but they don't. All right, so leakage channels. I have a lot of potassium leakage channels, meaning that potassium can pretty much come and go. Potassium moves very freely, for the most part, in a neuron. Told you we're going to get technical. I can have channels that only open if something binds to them. So here's my channel. And what we're saying is I had neurotransmitter or chemical bind to it, like at the neuromuscular junction, acetylcholine bound to it, and it would open up this channel. So it's ligand gated, meaning I have to bind to neurotransmitter in order for it to open. And then once that thing is gone, acetylcholine's gone, then the channel will close, right? So we saw that one already in muscle contractions. I can have mechanically gated channels, and mechanically gated channels respond to some sort of change in pressure or a vibration. So they're closed, but if there's some sort of mechanical stimulus like pressure, it's going to open and ions can flow in, but I cannot draw, that's weird, a straight line. That's crazy to me. Okay, so some sort of physical change, physical deformation has to occur in order for this channel to open. Another type, and I think this is the last type, yep, is a voltage-gated channel. And a voltage-gated channel will open when there's a change in the voltage difference. So when there's a change in like what ions are doing, it'll be like, oh yeah, I'm opening. So in this case, a voltage-gated channel, if the voltage inside a cell is negative 70 millivolts, we're going to talk about where that number comes from. If it changes to negative 50 millivolts, it opens. So at negative 70, and negative 70 millivolts is the resting membrane potential actual charge inside a cell. But when it changes to negative 50, so that means something happened, then this gate will open. So I had to change. I went from negative 70 to negative 50. And that happened due to ions moved across membrane because of something else. And you're going to kind of see this a little bit more in a minute. It's going to start to hurt. Okay. So if I'm a neuron, I can have leak channels, meaning they're pretty much always open. Sometimes they're closed and everything can flow. So these are mostly potassium. I can have channels that will open when they bind to a chemical like neurotransmitter. And these are more so sodium channels. I have mechanically gated channels that change in response to some physical change, a mechanical stimulus. And then voltage gated, which means they open and let ions flow because ions have already flowed because of another reason. So a change in voltage, a response in a voltage stimulus causes them to open. Kind of weird. All right, so at my resting membrane potential, let's get to this picture. When my neuron is resting, meaning it's not doing anything, think about my son laying on his bed watching YouTube all day. There's a lot of resting membrane potential going on there. I can take a crazy little electrode and zip it across and detect the voltage inside a cell. And inside a cell is negative 70 millivolts. Okay, so it's negative 70 millivolts inside the cell. Outside the cell, it's a lot more positive. I don't know what the number is outside the cell, so don't even ask me. Distribution, what is this one? All right, so we're going to tuck that away. And why is it negative 70 inside the cell? We're going to go to this one. And we're going to go to that other picture that I just flashed. But it's negative 70 inside the cell because I have more sodium outside the cell. I have an unequal distribution of ions. So I have sodium outside the cell. I have more potassium inside the cell. We're going to talk about why. Because potassium can go anywhere he wants to. Why is he staying inside the cell? I have other types of ions that are unable to leave the cell. And these ions are called anions. And anions are negatively charged ions. So negatively charged ions, actually proteins, charged proteins, are stuck inside the cell. And then I have a whole lot of sodium-potassium pumps, pumping sodium out and bringing potassium in. Okay, so let's look at this picture here. So let's start with the fact that I have the sodium-potassium pump, pumping out 3 sodium, so I have more plus on the outside, and bringing in 2 potassiums. Okay, so potassiums are these little triangles with the pluses. And there is my ATP. My ATP is driving the sodium-potassium pump. Why do I need oxygen and glucose constantly there? Because I am constantly running a bazillion sodium-potassium pumps, constantly pumping sodium out, and constantly bringing potassium in. So I have some sodium leak channels where some sodium can leak back in, not a lot, just a little. But what's going to happen is it's going to be grabbed by this guy and it's going to get pumped back out. Hey, sodium, get the heck out of here. What does sodium want to do? Because he's constantly being pumped out. Sodium wants to come into the cell, wants to come to the party. Because there's a ton of sodium outside and a ton of plus. Okay, so the outside is more positively charged. Oh, charge, right? And that's what these little pluses are meaning here. I have potassium leakage channels, and potassium can pretty much go out if he wants to. But you see, there's not a lot because potassium likes these negatively charged proteins. And it's like, oh, I like it here, right? Opposites attract. So potassium stays inside the cell. Because 1, it's negative. 2, no, because of the negative charge. Well, I'm just going to leave it at that. Stays inside mostly because of the negatively charged proteins are attracting it. Some will come out and they'll get grabbed and they'll come back in. And it's okay if it's out or in a little bit. But if you have too much potassium outside the cell, too much extracellular potassium, potassium won't flow right as it needs to, and things will stop working. So when people are using salt substitutes because they can't use sodium in their salt and they're using potassium chloride in their salt substitute, messing with their extracellular potassium levels, giving themselves heart attacks. All right, so factors contributing to polarization. Polarization is just a fancy name for saying unequal distribution. Sodium out, potassium in. Cytosol, so extracellular fluid is full of sodium. Inside the cell, full of potassium, full of proteins. And we already said potassium could do whatever it wants. The negatively charged guys are stuck in the cell and potassium likes them. Whoa, they're the bad boys. And I have the sodium-potassium pump that will pump the sodium out and bring the potassium in. And this guy uses tons of ATP. Therefore, I need tons of oxygen and glucose. I could talk about this stuff forever. It's actually kind of cool. I am getting to like these little rants. No, I'm not. So when I talk about a graded potential, remember those were just small little messages. And here I am resting, and I'm at 70, right? But I can become a little more negative. That's hyperpolarizing. What that would mean is like more plus has left the cell, right? More plus. Or I can become a little, so here I am at minus 70, my resting membrane potential. I became a little more positive. And that is called depolarizing. When I become positive, that's depolarizing. And that might have happened because, you know, more plus entered in. Come on, you. Maybe a little bit of sodium leaked in. I got to pump it back out. A little more potassium left. I got to pump it back in, right? So the sodium potassium pump will fix all things wrong with the resting membrane potential. And I'm just going to say RMP, resting membrane potential, all right? If you're like, what does that mean again? So I'm resting because sodium's on the outside, potassium's on the inside because he likes the negatively charged bad boy proteins. And I'm just ready, waiting for something to happen. So graded potentials are local. They're little, they don't travel. It could occur because I had a slight little physical deformation. There was a little bit of neurotransmitter, not enough. And some ions flew in or some ions left. Oh, binding of glycine binding. So here is like, oh, what the heck? Oh. My pen has gone crazy. Neurotransmitter opens up a channel. A little pressure opens up a channel and ions flow. So understand, I am resting at 70. I can have a little depolarizing. It gets fixed by sodium potassium pump. Obviously must be important because I keep talking about it a little bit more, but nothing happens. Your brain doesn't even know about this. This info doesn't go to the brain. Doesn't travel. It's local only. It's like between you and your neighbor, but nothing's going to another street, okay? I need to have a stimulus. Here's some stimuli. They're increasing in strength, you can see, because the little bar charts are getting taller. But it's not enough to really make an impulse. I still don't have an action potential, a nerve impulse. I need a stimulus that changes the inside to minus 50. That's my threshold. So a threshold for the real deal, an action potential, is for the inside of the cell to become minus 50. So enough ions have to flow to make the inside a change of 20. inside of cell. Whose job was it to figure that out? I mean, that just sounds dreadful. Needs to change from minus 70 millivolts to minus 50 millivolts. I mean, this is cool stuff. But, all right, so this is letting you know that gradient potentials can add. That's that summation. I can add, and I can get a little more. Let's go to the real deal. An action potential is all or none. It's either going to always be there and it's always the same, or it's not going to happen. But I have to hit threshold. And remember, that threshold was getting to minus 50. Oh, that's supposed to be a V. If I get to minus 50, then I will always have the same response. It will always be the same size. Always same response. Now, what's interesting is if I have super strong stimulus, right? I'm getting louder. How do I understand that the stimulus is stronger? Because it's not going to change it's not going to make my action potential any taller. So we'll come back to what it does. All right, so let's go into this slide first. Can I zoom in and write on it? I don't know. So there's 4 parts. Now, if I write, can you guys see it if I zoom out? Oops. I'm going to just leave it. Okay, so what is an action potential? And then we're going to go back to that other slide. Where's my pen? Okay. So the first part of an action potential is a resting state. When it's the resting state, I got to put an elbow up here, so hang on a second. I got to get comfortable. When it's a resting state, my sodium channels are closed. My potassium channels are closed, but there's leakage ones. All right. And now this sodium channel kind of has an interesting little thing. It's got the actual door there, but then it has this another little part to it that helps it stay open or closed. All right. So all of my gates are closed. It is minus 70 on the inside, right? That's what this is saying right here. So I'm minus 70. When the stimulus has caused it to get to minus 50, that's my threshold, my sodium gates will open. So sodium rushes into the cell. As sodium's rushing into the cell, it becomes more positive, all the way up to plus 30. So sodium goes in. That's the depolarization phase. Now, my potassium gates are still closed. So now, when these sodium gates are open, what's going on is this little ball and chain here, this inactivation gate, which is really slow, will eventually block it. It will close the sodium gate. It's not the right door that's closed. It's a separate little component that closes the sodium channel. So sodium can no longer flow into the cell. Sodium channel inactivation gates are closed. But potassium's like, oh no, you're not in here, are you? And I'm out of here. So potassium gates open up and potassium leaves the cell. So as potassium is leaving the cell, it's taking its plus charges, the cell is going to become more negative. That's repolarization. Now, what happens is, repolarization actually continues and potassium leaves more potassium leaves than should. So as it continues, oh brother, more K leaves, And it actually becomes even like more negative. Nope. Goes down below. And that's my action potential. I have the resting state. I have depolarization, repolarization, hyperpolarization. How do I get back to resting state? Because right now I've got all this sodium inside the cell, I have all the potassium outside the cell. How does it get fixed? It gets fixed by what? Yeah, the sodium-potassium pump. Fixes it so that it can do it again. Okay. So let's now go to, oh, no, this guy. Itch my nose. So this is just a graphic form of an action potential. Action potential is just a flow of sodium and potassium. It's really all it is. I will have the resting membrane potential where I'm resting. I will have a stimulus that is strong enough to go from minus 70 to minus 55. Once I hit that threshold, that level, I will always have the same response with the same amplitude. Same response, same amplitude every time. Now, that is all or none. Same amplitude, same response every time. Depolarizing phase, sodium enters cell. Sodium comes in because the gates are open. Repolarizing phase, potassium leaves the cell because his gates are now open and the sodium gates are now closed. But I will have hyperpolarizing phase, too much potassium leaves. And then how do I get back to resting membrane? My sodium-potassium pump. Oh my God. Now, I'm trying to write the Na in this little thing. Sodium-potassium pump will fixes it to be resting membrane again. Now, there's all these really faint colors. But what I want to point you to is this pink versus like this salmon. I have this part here. And this part here, well, even all before, is an absolute refractory period because it's going to go through what it wants to do. It's got to go through its process. Leave me alone. I'm doing my thing, right? When I'm the absolute refractory period, no matter how strong a stimulus, it will not fire off another action potential. No matter how strong. How strong stimulus the neuron will not fire an action potential. This other part here after it, when I'm kind of in my hyperpolarizing phase, is called my relative refractory period. A refractory period is a period of rest, and it's a relative refractory period because if I have a stronger stimulus, oh brother, will cause an action potential to be fired. Will fire. So what this is saying is that stimulus strength, if I'm louder, if it's heavier, stimulus strength isn't coded in how tall my response is, because my response is always the same. Stimulus strength is coded in frequency of action potential. So I'm saying that I'm louder, which means it's shooting off quicker. action potentials. All right, and that's what this diagram is saying. I have my, where's my pen? I have my stimulus. I'll have my action potential. Now, if I have this stimulus is stronger, right? When it's stronger, that isn't coded by making my action potentials taller, like muscle. It's coded by making it be more frequent, so it increased frequency. How cool is that? I think that's pretty cool. Makes me want to have another one of my Twizzlers, but I don't want to eat in front of you. Actually, I really do. I'm trying to control myself. Okay. So that was an awesome concept. I've been talking for 35 minutes. Let's see what's next. Or is it break time? Maybe I'll take a break. Oh, well, let's go through this. Okay, so refractory period. A refractory period is when it's a period of rest, when the neuron cannot generate another action potential, another impulse. Okay, so hopefully you're comfortable with the word action potential meaning impulse. I have absolute, even super strong, cannot generate it. And this is really due to the fact that the sodium inactivation channels, those little blobs, are still in the way. I got to fix everything and return it to a resting state. But I have a little bit later on in the process a relative refractory period where a very strong stimulus can initiate a second impulse. And that is how I code for stimulus strength. By tapping into this relative refractory period and increasing frequency. I'm trying to write the word frequency, and it's just not happening. Okay, increasing the frequency of action potentials. That's pretty cool. Oh, here it is again. So I have the absolute. This is untouchable all the way through to here. And then I have the relative. So if I had a really strong stimulus, then I would, shoot off another one. Oops. Let's see if I can actually draw. Nope. Shoot off another one and shoot off another one. You guys get what I'm trying to say on the most ridiculous thing. But otherwise, in this action potential, I have the depolarization, which it will always depolarize to plus 30 from minus 70, but it had to get to minus 55 in order to really kickstart the process. Oh, no. And then I have repolarization where potassium leaves. I'm not sticking around if sodium's in here. Hyperpolarization because too much potassium leaves. And then I have to fix everything to get back to my resting membrane potential. And that's my sodium potassium pump. So like we talked about in the last lecture, I didn't talk about it. An impulse can only go one direction, and it can only go one direction really because of those sodium gates. That inactivation gate, once it closes, takes some time to reopen. So once it happens, it can only go that direction because these are still closed, and then it can only go that direction because these are still closed. So that's why it only goes one direction. From the trigger zone to the terminal. Remember, the terminals are at the ends. The trigger zone's in the axon hillock. And these were the axon terminals. Okay, I think that's it. So once one area has had all this flow of ions, he's in the absolute refractory period. I guess I'm going to talk about this. I was going to save it for the next lecture. Let's stop here. I need a Twizzler break. And we're going to pick up here in a few minutes. All right, I'll see you guys.